Welcome to the Biotech Startups podcast by Exceeder. Join us as we speak with first-time founders, serial entrepreneurs, and experienced investors about the challenges and triumphs of running a biotech startup, from pre-seed to IPO, with your host, John Chi. In our last episode, we spoke with Quin Wills about his early years growing up in Johannesburg, the importance of seizing opportunities when they arise, and challenging the status quo. We also covered his early experiences in academia, including his time as a research scientist at UCL and his transition from medical school to studying genetic and computational biology, as well as his early exposure to and passion for bioinformatics. If you missed it, be sure to go back and give part 1 a listen. We continue our conversation in part 2, further discussing Quin's time at Cambridge, his pursuit of dual degrees in genomics and mathematics at Oxford University, and his early exploration of single-cell genomics, RNA, and gene regulation. We'll also cover his experience founding SimuGen during his graduate studies.
Eventually, I got accepted to the Oxford Master's Program and the Cambridge Master's Program at the same time, and ended up deciding to do both. In fact, the Cambridge one was more about traditional computational biology, learning to code, learning to think about big statistical modeling, learning to work with big data sets. Whereas the master's in Oxford was more traditional mathematics, applied and computational mathematics, a lot of mathematical biology. And so it was kind of 2 years of my life where I could just ignore the world and absorb as much as I can, not just about the science. I think what people forget is that while science might try to be objective, it's very driven by fashion about where we want to place our focus, right? And again, in the early days of bioinformatics, it was very unclear as to, the different philosophies or the different cultures that we're driving. So I was like, oh, what the heck, I'll just do both. Why not, avoid reality for another year or two?
Yeah, that's hilarious. And I think that is another thing that's worth reminding is math itself feels like this objective thing, but where you don't question it. And it kind of goes back to what you were saying before, where it's just that when you had that early classroom experience, you're just why is it being done this way? And during their time, in your master's program, if you're, jogging your memory back then, what were the paths bioinformatics? What was the fork or maybe multiple forks in the road at that point in time? And where were things going?
So I think for me, it boiled down to, do I want to do mathematical biology? And I think the answer to that was no. As much as I love mathematical biologists, they are incredible subspecies, but not for me. Do I want to do some of the more traditional, structural biology? And to be honest, again, not my thing. It came back to genomics, and again, I think that was my first big move away from traditional genetics, a DNA guy, to an RNA guy. And that's the point at which I became an RNA guy. And oh my goodness, we can, at those days it wasn't C. So again, let's go back to old technologies. This is how old I am. I was doing my MPhil in Cambridge when it was gene expression microarrays. We were printing things on glass. We were not sequencing.
Yes.
But I felt very strongly that this is a hook. This is a really interesting, chewy space to get into. And that kind of just drove, that was the problem then. And then that drove everything in my decision to do the PhD that I did, etc.
Very cool. Very cool. And so now, I guess before we move on to your PhD, during this master's, the dual masters, was it basically it was a dry lab? Or did you interface with wet labs as well?
It was very dry lab. And I think one of the things, and I learned this lesson hit me in the face quite hard during my PhD, is I hate analyzing other people's data.
Yes. Well, that's good that you discovered it, because
and the way I sort of explained, I had a very patient supervisor. And I was I did not do all these studies to analyze other people's data. I did all this to but again, it was a realization you have to come to, is that for me, it's about I'm very driven by, there is a problem out there that annoys me. I want to solve it. Data is a very big part of it, but there are many other elements that need to be solved too, and I want to be part of that. But kind of when we get to the story of Ochre, it's kind of reflected a little bit in that too.
Very cool. So, okay, so now you're heading over to Oxford for your PhD and in genomics. You've kind of started to pick your lane a little bit. Can you talk a little bit about your experience, your PhD experience specifically at Oxford?
Yeah, I had such a good time. I really, really did. It's PhDs are a slightly different beast in the UK. They're much shorter. I mean, I stretched mine out into 4 years, but they're quite short. So you just get on with it, right? Which I quite enjoyed. And I was surrounded by just incredible people, very inspirational people in this very intersectional space of biology and data. And again, got to see a lot of different phenotypes and the way they like to do things. But I think one of the biggest lessons for me was I do not like analyzing other people's data. I enjoyed it for a while because, when you're coming to a PhD, because again, you're nobody. Who are you? Here's a great data set, work with it. And I was very intrigued by it. And now it's old, but I was intrigued by the EQTL problem. It's a connecting DNA to molecular traits, connecting those. And again, you'll see this plays out in the Ochre story. But I thought that was a really, really fascinating area to be in. So I was very happy to be involved in big projects and be this very junior PhD student working on it. I just got very bored with it after a while because I didn't get the opportunity to structure the questions. I really wanted to get into single cell genomics. It was the very early days. This was even before anyone else, and really I'll explain how I thought about this because it actually came from my first company. Which I think we might get to in a little bit. But at that stage, I was really like, I think the right kind of resolution here, if we want to solve fundamental problems, need to be working at the fundamental units, cells, single cell, that kind of stuff. So there was a lot of that thinking going on. I was like, no, not this. I'm tired of all this EQTL stuff. I'm just going to. So I paid, again, such an idiot. I paid, I think I even paid for my own flight. I somehow got the money together. I paid for my flight, flew over to San Francisco, and somehow got myself introduced to Fluidigm, the very early single cell people.
Yeah. Wow.
And kind of made a pest of myself. I was like, I'm a PhD student at Oxford. I want to do stuff.
Yeah. That's the best.
And this was a very early, the single cell qPCR, but they had the technologies, the microfluidics to do big arrayed qPCR. So you could look at a lot of genes, right? It wasn't quite sequencing yet. And somehow I convinced my supervisor to go with this crazy plan. And that's, and I loved it. And it was exactly how I like to do science. And there's a problem. I see a technology I think really can solve it. And we really just chewed apart this idea of genetic noise and how it plays out. And, we still, and actually still today, we really think about gene regulation in very kind of Lego block mechanical ways. This binds to this or that. But there's a lot of control theory around noise in systems and sort of bursting or promoter bursting of genes and that kind of stuff. And this is stuff you can do with single cell. And so we just played with a lot of cute ideas, and I came out of my PhD feeling right. So, again, talking about science and then the phenotypes of the culture and how things work, it's amazing how different universities will have very different science, equally brilliant scientific universities will have very different science cultures. And there's often this comparison between the two Cambridges and Oxford, because Oxford's a quirky place. It is a very quirky place.
Yeah.
But there are a few things that Oxford does very well, and this embracing of wanting to solve clinical problems. If you think you can get hold of samples and new technologies and pair them up and think about a clinical problem, I think people are a lot more amenable to that, in my experience. And I think others have said the same. And so somehow I managed to convince people that I think this was the next step if you want to think about gene regulation and gene targeting and clinical problems, et cetera, et cetera, et cetera. And somehow they agreed and to shut me up, and we got a paper out of it.
That's awesome. That's actually awesome. Because I think, I love when you're talking about like these like different phenotypes in science.
Yeah.
And I think, especially for those who may not be, outside, who may be more outside looking in, might just be like scientists all come in one phenotype, but
Yeah.
there's just like such a diversity in approach, and even like institutions. And I was thinking about my time at Berkeley, and Berkeley has a very specific style of like, we're always compared to Stanford,
Yeah.
obviously for sports reasons and stuff like that, and just like historical norms, but like just such a very different place when it comes to like scientific entrepreneurship. And my time at Berkeley was just like pure academia, and like the concept of doing any, like, you're talking about Oxford embracing kind of like, if there's a potential for clinical impact, maybe let's go for it. And so at Berkeley, during my time there, it's different now. The concept of starting a business and spinning it out of academia was like shunned. They're just like, oh, it was bad. It was bad.
Yeah.
They're like, oh, you're going to the dark side. I kid you not. That's like me verbatim quoting it.
No,
like,
no.
not the dark side. Trying to help. I'm just trying to help.
No.
But the tides have turned, and it is a little bit more open now. But
it's such a disappointing. People forget how it wasn't that long ago that so many universities were like that. So Oxford was very much like that. Very much like that. For all the good.
Yeah.
You were made out to be a failure if that is what you wanted to do. And it's I think the thing, and again, this is personal experience. I'm always cautious to generalize too much, but having started my own company and being in pharma and now another company of my own, if I compare a lot of big academia and the politics of big academia versus my time, I find the science we do not only fast paced, but a lot more authentic because it just has to work. You don't have to hand wave and get a paper.
Yeah.
I find, at least in the genomic space, in industry, we're very collaborative. Very collaborative. I would argue more collaborative than a lot of academic setups. So I think I'm with you, but Oxford is slowly. There are a few really fantastic champions around Oxford that are also changing that narrative and getting people far more enthusiastic.
Yeah, and I love to see that. It's and it's not to pick sides either. Like I think there's
no.
Right? It's like there's room for it all. And it doesn't have to be antagonistic. It could be like, can we work together on this? And that I think is, it kind of just, it always was previously kind of like butting heads, but it's just like, it feels far more collaborative now. But yeah, this kind of dovetails nicely. So I know you started a business during your graduate studies. It's so SimuGen. Am I pronouncing that correctly?
SimuGen, yes.
Can you talk a little bit about that? Like, one, did you sleep at all? Like, how did you? Okay, yeah. So can you talk a little bit about the business you started in grad school?
Yeah, this is why I want to sleep now when it comes to New Year's.
Exactly. Again, come to the dark side.
I'm old and worn out.
Yeah.
Yeah, no, so this is why my supervisor was an absolute saint, my PhD supervisor, because I'd started this SimuGen, co-founded it, coming out of my master's program. And I was due to start my PhD and sent my supervisors, or both of them, I sent them an email, this time from my own laptop. Internet cafe. And was like, can I start my PhD 6 months later? And I could already tell both of them were like, oh, this one's going to be a spicy problem. Yeah. But it was fine. I mean, it made no real difference to them, And so I did a lot of SimuGen during my PhD, did a lot of things in chunks, so I'm a big chunk. I can't multitask. I have to focus on things in moments. So I spent 6 months really getting a lot of the up and going. And then at points in my PhD, there were times it was, it's all right, I'm going to spend 2 weeks in Malaysia now working with the computational team out there. So then I'll come back, carry on a PhD, and then I'll do 2 weeks here. And, I do kind of a little bit like my life is right now. I can't imagine being any other way. I really love it. It's really fun, engaging lots of different teams from all over, getting on with it, moving fast with stuff. No regrets when it came to SimuGen.
And can you talk about were you the sole founder? Did you have like a co-founder? And like, what did SimuGen does?
Yeah. No, so it was co-founded, and the idea was I came with this idea that, again, I wanted scientific, well, founding CEO, but then became CSO, of SimuGen, and so it brought this idea that, okay, we want to do liver. Liver tox is a big issue, and it still is in drug development. People are, looking for earlier stage models, and this was when high content imaging was becoming really, really vogue, and everyone was like, "Wow!" And I was like, "What? I could do better than that with gene expression arrays." And the whole idea was to either, like a lot of things, the idea evolves, but the idea was this was going to be either in parallel with or replacement in many cases for high content imaging. And it was the same idea. You take some models, but instead of just imaging, you put the stuff on arrays and so you're getting mechanistic information and hopefully making better predictions of tox endpoints than imaging. And that really was the idea, I think, but there were many, many great lessons in SimuGen, not least of which was, you can have a great idea, but if the market doesn't want it, good luck.
Yep.
Which, like, people were not prepared to pay for it. I mean, gene expression arrays were new tech, and they're like, "Why? How much?
Yeah.
I could do this for 2 cents with a microscope."
Yeah.
And a few clever stains, and it was a really great sort of wake up call in terms of how, and you say for business, but in life, really too, right? Even in developing therapies, in the liver space, there are a lot of areas like NASH. Will people pay for it? It's just, you can create this incredible therapy to reduce fat in the liver. Will they pay for it, and how much? And so there was a lot of that, and there was a very big lesson in product market fit.
Yeah,
starting a company.
That's amazing. And so there's so many directions I kind of want to go with this, but was this your first time you tried your hand at sales? It's like where you're starting to like talk to the market, interface with like potential customers.
Yes.
How was that experience?
You should probably ask the people who worked for me what that experience.
Okay.
What's it like working for a first-time scientific founder? And you'll get some real horror stories. No, I mean, I'm you can't tell, but the lighting is making me look so red, but I'm blushing every time I say stuff like this. But I made all the classic mistakes. I really, really did. The product, not paying enough attention to product market fits, and just clever tech, big mistake. And another one is I was very driven to make this work. And I think one thing when I left SimuGen, I deeply regretted certain things I said and did. Not that I unethical or, I think what a lot of first-time scientific founders forget is that science is 10% tech, 90% people. And you have to spend a lot more time in empowering. And I mean, I'm still sucking at it, so don't pretend that I'm lecturing, but I try much harder on that element, and to really focus on the people side of things. And that, I think, was a very, very big lesson for me coming out of SimuGen.
That resonates so much with me, because I think exactly, you're nailing a bunch of concepts that I think aren't, for whatever reason, isn't really touched upon much in it always, the saying, like, if the science is sound, it all works itself out.
Yeah.
But if you can't, right, I'm like, but we sometimes can easily forget that who's driving the science. It's a group of people. Like, it can quickly turn if the people element isn't right. And exactly, we're getting back to like, science can feel objective.
Yeah.
But it's far more multivariate and far more subjective than we would like to tell ourselves. And also another thing too, in addition to people, the concept of product market fit in kind of like science, it's kind of like people just assume that there's already product market fit, like coming out of the gate, that it's already done. But like, that can be like a brutal reality where you pile a bunch of, you just muscle through it, you white knuckle through it. You're just like, we're going to make this work.
Yeah.
And then you're so proud of the science and the product. And then when you bring it to the market, and it's just like, nope. There's like kind of like I mean, I absolutely took it personally. Early days, that was a hard lesson learned.
It's very tough, and that sort of a slight tangent to that same lesson is that just because something's valuable doesn't mean it can be monetized. And I think this was a big lesson a lot of us learned in what I call the first wave of bioinformatics. Now we're in the second wave of bioinformatics, and everyone seems to have forgotten all the lessons we learned in the first wave. Because everyone's like, we're going to generate lots of data. It's going to be valuable. I'm like, oh, how's that business model coming along? How much are people paying for your data? People want assets. People want de-risk assets, And it's just that having just a clearer vision for what that journey could be at the start, I think, is a far better investment in your time than having a perfectly formed idea. Because scientists always want we need a brilliant idea to start it. It doesn't need to be brilliant. You just need to see what the journeys could be, and you'll already be doing far better than everybody else.
Also, I think too, just like being willing to adjust on the fly. I think being a lot of the time too, that like you can easily get wedded to your idea, your initial idea.
Yes.
And that can just blind you to exactly what you're saying, the multiple paths that are in front of you. And I'll say I'm a pretty stubborn person, so this is not to I'm speaking for myself. Like if I look back at my past, I'm like, John, you probably should have kind of pivoted a little bit earlier, and it probably would have been far less painful. But I think those lessons too, kind of are lessons best learned firsthand. Like it's one thing to hear someone say like, yeah, be open to change, and the market might not like what you have to offer. You're like, yeah, they're gonna like what I have to offer, but until you it's kind of one of the sayings I have internally, again, kind of this broken record thing, just like, I can tell you how hot fire is, but you don't truly know until you touch it yourself.
Yeah.
On paper it's hot. I can tell you that, but you don't really know.
No, there's no, and I think a lot of us thought about this when we were much, much younger. I thought like this when we were much younger. I definitely felt like, you can think your way through anything. Why, this whole thing of life lessons, if you're just smart, and it's like, no. And here's why. When you're dealing with complex systems where there's not a tidy equation, there are so many feedback mechanisms, so much happening, that the only way to learn and get your internal AI to recognize these things and respond at the correct time is to go through it yourself. It has to be learning by experience.
That's all for this episode of the Biotech Startups podcast. We hope you enjoyed our conversation with Quin Wills. Be sure to tune in for part 3 of our conversation to learn more about his journey. If you enjoyed this episode, please subscribe, leave us a review, and share it with your friends. Thanks for listening, and we look forward to having you join us again on the Biotech Startups podcast for part 3 of Quin's story. The Biotech Startups podcast is produced by Exceeder. Don't want to miss an episode? Search for the Biotech Startups podcast wherever you get your podcasts and click subscribe. Exceeder provides research labs with equipment leases on founder-friendly terms to support paths to exceptional outcomes. To learn more, visit our website www.exceeder.com. On behalf of the team here at Exceeder, thanks for listening. The Biotech Startups podcast provides general insights into the life science sector through the experiences of its guests. The use of information on this podcast or materials linked from the podcast is at the user's own risk. The views expressed by the participants are their own and are not the views of Exceeder or sponsors. No reference to any product, service, or company in the podcast is an endorsement by Exceeder or its guests.